Then let's start. So good afternoon, everyone. Welcome to the second installment of the October mini-school on empowering biodiversity monitoring to data cubes. And we have again Dr. Sandra MacFadyen and Maarten Trekels, who will sort of share the lecture again. Yeah, I don't need to introduce them again to you because you saw them already last week. Yeah, the slides of last week are on the NITheCS website, so you're welcome to download them from there and study them again. Yeah. And probably after today, we will upload the second set of lecture notes of slides on the same website. Yeah, Sandra, if I understood it correctly, you are kicking off,
Yes.
then passing to Maarten just now. Yeah.
That's
Okay.
right.
So thank you very much to both of you. Yeah. Thank you very much. And yeah, I'm looking forward to the lecture. Thank you.
Thank you, Francesco. Thank you. And yeah, welcome back everyone to our mini school. I'm Sandra MacFadyen, as Francesco said, and joined by Maarten Trekels. We won't go through that again. And we're going to start our second lecture today related to biodiversity monitoring using data cubes. And just to give you a quick run through again of the proposed outline, what we did last time, we did a basic introduction to biodiversity and the concept of data cubes. We looked at different sources of biodiversity data and reviewed some of the data standards that are needed to be in place for us to start creating data cubes. We also touched briefly on data publication and fair data publishing principles. And we gave you some homework to do. So some of you have hopefully linked up your different accounts. I'm going to give you a slide in a minute so you can quickly do it if some of you haven't finished it. Because today, Maarten's going to run us through session 2, which is actually building data cubes using GBIF. And he's going to take you through an introduction to what is GBIF data, and then you're going to do a hands-on tutorial actually coding on looking at biodiversity monitoring. The next session, I'll take you through some ecological modeling with data cubes, looking at also creating your own data cube using GBIF, Google Colab, and Earth Engine. We're going to look at how do you visualize your results on an interactive map and how to analyze some patterns related to species richness and environmental variables like climate. Our last session will be a session on open science, looking at how you share and disseminate your results. And we'll do a bit of a hands-on session with GitHub and Zenodo to show you how to do that. And we'll end off with what do you want to see next? So we want to hear from you. And so think about that in the meantime. This is the accounts that you needed to set up for today's session. So I'll leave this on the screen while you guys quickly do a mad rush to create the accounts if you haven't done that. We'll be doing some hands-on stuff with GBIF, Google Earth Engine, and Google Colab. All right, so I'm going to hand over to Maarten now. I'm going to stop sharing my screen so that he can take over.
Thank you very much. And this will be a session where I will try to do go live through all the data steps. So it's a bit of a live session, so things could go wrong. Sorry for that. That's just how it works. But I will try to go through the steps slowly. If you have, in the meantime, any questions or things that are unclear, or whether I go too fast or too slow, please let me know in the chat. You can always ask questions or give suggestions or ask for clarification. Sandra will look at the chat and look at your messages, and then we can address them during the lecture. Don't hesitate, because you might have forgotten your questions by the time we get to the end of the session. I will start sharing my screen. Yes. Okay. Can you see my screen? Yes. Okay. So basically, I will go step by step, as Sandra already said, and we will start by going to what the GBIF data is, because we are going to build GBIF data cubes, and to do that, we at least have to understand how GBIF data looks like and how we can access this data. So if you go to the homepage of GBIF, gbif.org, I'm not sure you can you see my screen? Is it clear?
Yes, we can see it.
Okay. You end up on this homepage. And on this homepage, you can already see that GBIF is an integrated and data integrator, as we spoke about last week. And so you have different kind of views on this data. One of the views is you can look at it per data set. So if you click on data sets, you get a list of all the data providers that are providing data to GBIF. And for example, the eBird observation data set comes first because it's contributing a lot of occurrences to GBIF. So that's one of the different views that you can have. If you click on a specific data set, what do you get here? Well, you can see how it's spatially distributed, all the observations that you get, how many there are, how good they are matched with the GBIF taxonomic backbone, how many are there with coordinates, and so on. Alongside with some description on what this data is, how it's collected, and which countries are it's collected in. So you get a whole bunch of metadata around this data set. So if you want to know more about specific data, this is a good place to look at it. Alongside of this view, you can have some metrics. The metrics are, for example, taxonomically, how these are taxonomically organized. As you know, these are birds, so they will probably all fall into the Aves. And how it's collected over time. And you see that eBird is starting to really kick off somewhere around 2010, 2012, and that you have an exponential increase in a number of observations. So this is one of the views. If we go back to the homepage, you can also look at species. So what kind of species are there in GBIF? And if we just select a single species, I'm just taking something random. You get all the species that are matching with your search that you did, and you can have a look on what kind of information that you get on these. So basically, you can see for this species, it's at the level of a variety, it's an accepted name, with some images, if there are images on GBIF, again, a sort of distribution map, and where all the information is coming from. But today, we're not going to focus on the dataset view or on the species view. They are important, but this will not be the main focus of today. Today, we are going to look at the single occurrences. Any single observation or specimen or whatever we can find in the GBIF dataset. So if you click on occurrences, you will get to a list of occurrences. In principle, if you don't put any filters on them, these are all occurrences. And right now, these are more than 3 billion results of occurrences that are on GBIF. So, of course, you have to start filtering. And all the filters that you can apply are actually here on the left-hand side. It gives you a subset of those. But if you click on all filters, you can see there are a lot of terms you can use to filter your data on. Also here, we will first do a filter. Just I will take again scientific name. As one of the filters. then we'll at least narrow down already a bit all the occurrences that we get here. And we can also, for example, try to limit our search by country. Let's take Spain, for example. And then you get all the lists of this species, all the occurrences of this species in Spain. Also, in this kind of view, you still can have summaries of what you see. If you go to the metrics tab, you can at least get an idea when these occurrences are observed. For example, you see a peak in observing of this species around April. You can also see a temporal distribution of when this species is observed in Spain. Be careful, we are looking here at a filtered view, so we are only looking at the ones in Spain. You can see the current filter here on the top. You see when they were observed. Basis of record. So basically the yellow here is human observations. This is typically something which is observed through citizen science platforms like iNaturalist or observations.org. But there are also preserved specimens. These are occurrences or specimens that were collected and are somewhere held in a natural history collection. And then some that are not specified as occurrences. You see that there are no fossil specimens in this case, no material citations. So material citations is a special case where you actually have a digitized literature, for example, or also no living specimens in our dataset now. Also, there are some issues. So not all of the data, so GBIF does do some data quality checks on the data that is published and does notice that there are some issues with data, for example, coordinates that appear to be rounded. And so all of these issues per occurrences are flagged, and you do get an overview of what type of issues you can expect with your data. And then let's go back to the list. So here, or you also have a gallery. So if there are images available, and most of the times from citizen science platforms, you do get images of your species, or if from the occurrences, you can see them, or the map, how are they distributed. So basically, since I selected on Spain, you do expect them only to appear in Spain, which is, of course, logical. If we go back to the data table, we can now just zoom in to one of these occurrences, because we do need to understand how the underlying data for each of these occurrences look like. Just simply click on one. And what do we get here? Well, again, you get the taxonomy or the scientific name in this case on the top of a single occurrence. You get some vernacular names, so local names. It does know that I'm in a Flemish-speaking part of Belgium, so it gives me the vernacular name in Dutch, and it gives some taxonomic information here. In the second part, it gives you who published it in what kind of data set and a reference to that observation. It gives you a location. If there are images, the images are popping up. And then actually most of the data is below here under in this section. And this section is split up in several subsections. So you have information on the record level. This is very general at the highest level, what kind of observation it is, in what data set. Then you get information on the occurrence, which is typically holding an identifier for that occurrence and a recorder, who recorded this occurrence. Information on the event, so when and where this observation was made. Identification, who did the identification, and some taxonomy. detailed information on location, and maybe some other information such as the license, which is attached to this observation. And maybe some references, for example, in this case, multimedia references to the images that were taken. So straight to the iNaturalist website, but this could also point to a website from the collection if it's a specimen. Okay, good. This is basically basic information, and all of these fields that you here can see in one occurrence are searchable. So the filters that we saw in the beginning in GBIF, they are actually derived from here. So if I filtered on country Spain, this is basically one of the data fields which is available, and all of these data fields are searchable. So having an idea how a single record looks like is definitely important to keep in mind. How do I now access this data? So this is, it's nice to see this data on a website and it gives you a very quick ID. But if we go back to our list of filters that we did, and actually this is something anyone can do. So basically you can do filters and make a single view with a certain amount of results. If you then want to do further analysis on these results, you can actually download this as a data set. If you press the download button, you can have a few choices here. You can have it simple as a Darwin Core archive or as a species list. And depending on what you take, the one contains more information than the other. For example, the simple file gives you only the interpreted data by GBIF, while if you download a Darwin Core archive, which is basically a fancy zip file that contains a CSV with the data, also contains the raw data. So when I told you that there are sometimes issues flagged by GBIF, sometimes this data is corrected by GBIF or is omitted. But if you then want to go back and look at the raw data, you need to start off with the Darwin Core archive. It gives you a few basic information on this. If you are happy with this, I'm just gonna, for simplicity, take the smallest size of data file. So I take the simple file. You will get some information, the fact that you have to agree with the user agreement of GBIF, but for the rest, it's free of any costs. From the moment that you make this download, and in principle this should have worked, yes, it will start processing your download. So basically it's not something you will be able to download immediately from the website. You will have to wait until in the backend of GBIF this dataset is created, and when it's ready, you will receive an email for this dataset. And then you can go back to your user account where you will see all the downloads that you requested. Why is this important? From the moment that you create a dataset and you want to download it, it will create a DOI, a unique identifier for this type of dataset. So at this moment, you have full provenance of where this data is coming from through the link with this DOI. And it does give you the filters that you used when you did the download and so on. Whenever your download is ready, and we're not going to wait until this one is ready, but you can go to your own user profile, go to downloads, and find all the downloads that you ever did before. And then I will just take a simple one, this one, for example. You will have a download button here, and you can click on it, and it will you can download this file to your own computer. Okay. From then on, you can continue with any kind of data analysis that you want to do on this specific data set. That's one way of accessing the data. Another way, I'm not sure, are there any questions, urging questions at the moment now, because this is still very basic, and I assume that a lot of you already know about GBIF data and how to use GBIF data. But if not, if something is unclear, let me know in the chat or raise your hand and we can listen. Okay, if not, I will continue. This is one mode of access to GBIF data. Another mode of access to the GBIF data is actually going straight to the API. So API of Application Programming Interface, if we go to the website of GBIF, the GBIF API, then you can see that you can also programmatically access all this data. Again, according to, and maybe my screen is a bit small, I will try to make it bigger. Yes. You can see all the things that you saw on the homepage of GBIF, the occurrences, species, the registry, which is actually the place where you register your dataset, are accessible straight from the API. As an example, I will just simply go to the occurrence API because we are mainly interested here in the occurrences. And you will get this kind of information. And this, for people that are not familiar with APIs, that this might seem a bit complicated, but actually it's not. It's not so complicated, and it's actually quite easy to access. Basically, it tells you on what kind of server endpoint you will have to request your data. So not straight from the GBIF website, but now on a subdomain called api.gbif.org version 1, because that's version 1 of the API. At that moment, you can actually submit any kind of request to this server. And the basic request, I'm not going into detail of all of the possibilities you can do with occurrences. You can search, you can get, you can download occurrences like this. But for example, this one, which is the most simple one, to get an occurrence, you need the GBIF ID. For example, if you want to get information on a certain occurrence, and if we go back to our GBIF website, this is a feature that I didn't mention yet, but if you take any random occurrence here, like this one, you will see that this occurrence has an identifier. It's displayed here in the URL to that occurrence, but you can also find it back in the data itself. I'm not going to search for it now, but in principle, this identifier, that's the one that is pointing to this occurrence. What happens now if we take this identifier and send it to the API? So we just gonna go straight back to the API. The syntax is explained here. So you have to have the link to the endpoint / occurrence / ID. So we're gonna do this occurrence / ID. And we click on this. And what you get out of this, and I'm gonna increase it again, is structured data, in this case in JSON format. And in this structured data, you will get all the information that you can see from the occurrence itself. I already closed it, so but you can see here things like the scientific name, the accepted scientific name, some taxonomic information, some information on where it's located, the year of the event information, but also identifiers, in what country it was collected, and the GBIF ID, of course, as well. So basically, all the information that you now could see or access through the interface of GBIF, or the information that you get by downloading the data, you can also programmatically access in a machine-readable format to the API. So you don't necessarily need to download all the information in a single file. You can send requests to this endpoint on single occurrences. Moreover, there are software packages in Python and R that are providing you that access to that data, so that you don't have to, in your programmatic language, if you are programming, not always have to send the same kind of syntax over and over again to that endpoint. There are software packages written around it that make your life a little bit easier. And one of those is BIOGRAPHIES. This is the Python package that gives you access to the data. And if you go to the GitHub repository where BIOGRAPHIES is developed, you will see all the information here, but also how you install it. And there are some examples on how you use it. Let me just quickly check whether I'm missing something here. I don't think so. Okay. Another way of accessing this data, and let's go back a little bit here, are having API downloads. So basically, all the filters that you saw in the beginning when I was filtering on all the specimens in Spain, you can actually also send to the API in a JSON format. And let me quickly check if I can show this to you. So basically, you have to I have to quickly check now. Oh. So if you, I showed you how to use the API. There's some more information here on the Python and R packages, but there is a third way, and this is the API downloads. I will mention this now because actually we need the syntax of this API downloads to access our data cubes. So when we are going to create our data cubes, we will do this through this API downloads. It's very similar. It goes exactly the same way as we did before with the simple queries to the API, only by, instead of sending a single string to the API, we are now sending our search term into a JSON format. For example, you create a JSON file which is called query.json with your username and email address. And then you actually say, okay, I want a simple CSV file or a Darwin Core archive. So you can choose the format here. And the query that I want to send to it is it needs to be a preserved specimen and in one of those two countries. So basically, this gives you a way to send programmatically a query to GBIF and get your data back. The way you do that is here. The only caveat here is that, again, this will initiate a download at GBIF site. So it's not immediately at your disposal. It will generate your data set in a way that it's looking at your JSON file. But then when it's done, you will again get an email saying, oh, the file you have is there. And I think now is actually a good moment to just practically show already a little bit. So for this lecture, I created a Jupyter notebook, and I just want to, before we go into this, want to ask: is everyone happy with your homework? Did it work? If not, please ask it in the chat that we can have a look at it, why it didn't work. But the first thing is the Jupyter notebook. You can actually download this Jupyter notebook yourself by going to the GitHub repository from our mini-school. So I will paste it in the chat. Here, if you go there, you will end up in on this repository, and there is a link here at the top which says Open in Colab. When you click on this link, it should open this notebook into Google Colab. And if you are ready with the homework, you can do so, and you will end up in a Jupyter notebook like this. How does it work? This is now done in Python, so you see a lot of Python code here. I will not go over all the details, but sometimes there are missing packages that you have to install. So for example, the Google Colab notebooks don't have PyGBIF installed as one of the standard packages installed on it. So basically you do have to install that package first. And then you have to import those packages or load the packages that you need. How do you do that? So you see that there are blocks here of text and blocks of code. The text is free text, so you can modify this yourself if you want. So you can just type something and it will add to the text here. It's Markdown, so it should be quite straightforward. The only things you need to know is that the pound sign or the hash is just giving you the fact that it's a title, and so one hash is a higher title than three hashes. How do you, then you see the blocks of code. How do you run the blocks of code? You see this play button on the left. So when you click on this play button, and I will do it here, it will run. And when it's finished, you will get a green V at the left-hand side saying that it worked. If it didn't work, it will show up red, so you will know when you are not there. I'm gonna quickly skip over the Earth Engine. We will get to that later. But here, I want you to have a look on how the GBIF accessing to the GBIF API looks like. So in this case, we are using the PyGBIF package. And what I want to do is I want to import the occurrence module in this. I want to see the occurrence view of PyGBIF. And I want to request everything. And that's something I probably forgot to say. If you go back to the API, let me just quickly show it again. You will see in the API that you have a species, which is just a species name. But basically, GBIF is not working with the GBIF with the names itself. But for each species, you will get a key. And this key is a number. And by requesting this number, you will get all the occurrences of that key. So just as an example, we can copy the species key for Pika Pika, and then we can see, we can send this here by saying I want, oh, it's the taxon key in this case, but it doesn't need to be the taxon key, so I can also look for species key here, and I paste the number of the species key, so I will get all occurrences with species key, this one, which will be Pika Pika, and I will limit it to 10 to make sure that our data set is not too big. And so what you get here, if you run this code, is the first 10 results of looking for the magpie, basically. And this is also in JSON format. So if you want to actually get some information on this, you can look for in this data structure. So here you actually straight from GBIF requesting data for magpies in this case. Okay. Any questions on this? If not, just please don't hesitate to interrupt or ask, and we can have a chat. Okay, good. That's so far for GBIF data. At the moment, we are still not working with cubes. So if you want, our session is called Data Cubes. So basically what we're now going to do is, from the basic understanding of what GBIF data is, create a data cube from this data. This work is partly performed in the BCube project, which we mentioned last time. But the information on how to create a data cube is also available on the GBIF website. And if you go to the technical documents of GBIF, you can see here in the API downloads. So it's a special version of the API downloads. You can see here species occurrence cubes. And what is a species occurrence cube? Basically, what we are going to do in the species occurrence cube is make sure that we are already doing some basic data cleaning and aggregation before we are playing around with the data. In many cases, you don't need all the information at a single individual occurrence level. So by creating these data cubes, you will already aggregate according to a specific format that you want all the data in a single file. And we are just going to look at how the standard cube would look like. And basically, as we said last week, the most simple cube format is a data cube in 3 dimensions, which is time, space, and species. So basically, what kind of species was where at which time. And to do that, and I'm going to straight go to the full query. Here, we are aggregating the whole set that we want to look at in these different axes of your data cube. In the dimensions that are going to be used here are maybe a bit more than what I explained now, but don't worry, it doesn't matter. You can actually choose yourself what are the axes of your data cube. And the first one is time. So basically, how do you create a GBIF data cube? And this is using the SQL query language. If you're not super familiar with SQL, that's not a big problem. Some of the examples that are given online and the example I give in the Google Colab section are relatively easy to understand. So basically, you want in this select statement, in the first thing that you do is there you define the axis that you want in your data cube. And the first axis is time. In this case, time is an aggregation of year and month. So the granularity of the data cube that you are generating here is a monthly cube with as a name for the timestamp year month. This is again something you can choose. You can do it on a daily basis, monthly basis, of even yearly basis if you really want just coarse timestamps of your data. This really depends on what you want to do. The example here takes monthly steps because it's something in between, and it shows you how you can do this. Then there is a spatial component, and for that, GBIF created a number of functions. In this case, this function that you see here is a function which is creating EEA grid cell codes. The EEA grid is a European grid system that divides the European Union in a grid, but you can use others. So you have the military grid if you want to use it, but also quarter degree grid cells. And in the collab session, we will check out the quarter degree grid cells. And then you have species, also family and species key and so on, but we don't, not gonna look at it now. Then you have to select from where you want it. So we are looking again into the occurrence view. And then there is a statement that says what kind of filters do you want to apply to this? And this is actually the data cleaning step. So I want everything which is older, every occurrence is after 2000 from Poland, and it needs to have a status present. And there are a set of other requirements that your data has to have before being included in your dataset. And then you group it by all the axes, and then you can order them either descending or ascending what you want. And that's actually the basic thing that you have to do to set up your species cube. When you have your SQL query, similar to the other API downloads, you have to send this to the GBIF API by making this kind of JSON file, which is the same kind of JSON file that it has to be sent to this email address in what kind of format. And we are looking at the SQL TSV zip files, which is our data cube, the query you want to send. And if you send this to the API, you will create a download which is your data cube. To speed up a little bit, I already pre-generated some cubes in this data folder, which you probably can get access to. So I can share this link with you. And in this link, you will see that I already pre-generated a few cubes. For example, here I did a cube on South Africa on quarter degree grid cells at level 3, which gives you about, I think, 1 kilometer or 10 kilometer grid cells. Okay, good. Then we can actually go and have a look on how this data looks like. Let's have a look. This section here is how to download it from your Google Drive. Basically, if you take the files that I sent in the Google Drive here and you put it on your own Google Drive, you can mount your drive and read the data directly from there. This is by using these two steps here. And what this will do is mount your Google Drive in Colab. And then if you click here, you can actually navigate through your Google Drive and see where you stored your file. That will give you a link, and that link you paste here where you have to read the CSV as a data frame. So basically, if you want to look at now you can see my whole Google Drive, of course, but you have to put that file somewhere on your Google Drive, and by mounting that drive, you can access that data straight from your Google Drive. Okay, good. So I've read it in as a data frame, and you can have a look at it. I also loaded in that same drive, and you can find that file here as well. the quarter degree grid cells. So basically these are stored as geo packages. So you can use those as a sort of shape file or a geographic file to load your data. How does it look like? It's also a data frame, but the data frame contains the name of the grid cell and then the polygon that is delineating that grid cell. Of course, in this one, and I can show you if you want to add extra lines of code, you can here do plus code and you get a new line. You can have a look at your data. This is the data which is coming from GBIF. It gives you, again, this year, month, like we said, the grid cell code, and then family keys and all the other access that you defined in your query. You have to merge this to get also the geometry, which is just getting two matrices together. So in this case, if you merge it, you get this merged file, which gets the grid cell code, your species, how many of them, and then the geometry of your grid cell. We converted to a geodata frame to be able to work in it in Google Earth Engine. And of course, this is for all, I generated this for all species in South Africa. So if you want to look at a single species, you can, on this data frame, put filters on and say, I want to look at everything with species key and then equal to a number. In this case, I was looking at elephants. So I looked up the species key of elephants, and then you filter your data according to that axis. That's very useful actually, because you can actually slice your data cube according to any axis. So I want everything of September 2024, which is also a good way to slice your cube, or whatever, the way you want to look at this. Okay. I see that I'm taking quite a bit of time here, so we still have to go to the Google Earth Engine, but don't worry, we will come back to that also next week. But I will show you a little bit here. We are also using Google Earth Engine here, which is, if we go here, a platform, a Google platform that contains a lot of data sets, and the data sets are climate data, satellite images that you can combine with your data cubes. If you want to, for example, look at surface temperature or any kind of climate data according in relation to your species, you can go and look whether you can find a data set here. And you can access this data set either to their editor, and I think Sandra will cover this a little bit next week, but also through Google Colab. So basically, if you go to the code editor, you can you see here how you can access your data sets through to code. So you can do this is JavaScript, not Python, unfortunately. You can access these data like this. We are going to do the same, but now using straight from Google Colab. So I want to look here at the Landsat data, which is imagery, the boundaries of countries, and then this one here is the World Database of Protected Areas. So these are 3 data sets I want to load in from Google Earth Engine. One little caveat here is that to authenticate Google Earth Engine, so you have to link your Google account or to log into your Google account to access Google Earth Engine. From last month, I think, you have to have a project attached to Google Earth Engine. This is not so difficult, and so if you go to the Google Cloud Console, you will end up here, and you will have to, on the top left here, create a project with a name, and that project should be allowed to use Google Earth Engine. So if you go here I loaded this project now here, and you can see here with API and services, you had to enable Google Earth Engine API to access this project. That's just when you want to have a paid service by Google. So everything I show now is free, but if you want more compute power or more data storage on your Google account, you have to use paid services, and therefore you need to link up to your project, and to make this uniform, Google now asks you to load it anyway, and you have to enable this API on the top. But it's quite self-explanatory. Basically, so I imported here in this, and you can play with this. So please use the Google Colab and play with this. I loaded this dataset and clipped it to South Africa. And what you can do then now is, so if you remember well, I filtered on elephants. I can now map them on a map. Here, for example, I loaded the three layers. So the Landsat data are the imagery. So this the dark thing you see on top of it is the Landsat data. I can disable this just to give you a better view on what it is. The black polygons are the protected areas that I loaded in. So this is the World Database of Protected Areas. I don't want to use it for now, so I can click it away. And so here you can see the data cubes, which is containing the elephant data that I filtered before on the quarter-degree grid cell grid for South Africa. Well, there are a few, and I have to look into this, why there are a few in Botswana. I don't know why, but you can see here nicely this data set. And this gives you a lot of possibilities, and I will not go into detail this, but now you can start playing with the grid cells itself. So if I want to derive something on climate and you have a climate layer, you can take this grid cell, clip it to the climate data and get information on the climate data. Or in case of the protected areas, you can count now, for example, how many of your data cells on elephants are within a protected area and how many are outside of a protected area. So now you can start playing with the simple GIS, or simple, it's not simple, but now you can do GIS on this data and get additional information out of your data cubes. And I think with this, I will keep it for today. I had some other material lying around. You can look at it later, but please go ahead, play with this Colab notebook, and come back to me if you have any questions or need help with generating the cubes. Thank you very much, and I think we can take some questions now.
Yeah, thank you very much, Maarten. I think there were a few furious message exchanges in the chat, but I think Sandra took care of all of them, if I see it correctly. If there are questions, please, you're welcome to raise your hand, and I can give you immediately the right to ask the question in person. Otherwise, you're still welcome to use the chat or the Q&A. I think Sandra is monitoring them both. Yeah. Okay, there are lots of thumbs up coming up the screen.
I have a quick question for everyone online.
Yeah, please, Sandra.
Please let us know in the chat what the speed is like for you, if we need to speed up or slow down, so that we can adjust the lesson next week if necessary. So just put in the chat quickly if the speed is perfect, if you'd like us to slow down, or if it's a bit too slow and we need to speed it up. Thank you.
It was okay for me. Yeah. But let's see what comments. Speed is good, thank you, they say. Little bit more time between steps. Speed is perfect. Yeah. Okay. Maarten, you did a great job. Yeah.
Yes, and I
Maybe
thought
some,
I
maybe
had not
just
enough.
some reading time in every now and then for people to catch up. But I think it was just perfect. Yeah, no, thank you very much. That was really very nice. So, and those are all the comments that we have now. Yeah. Okay, one I would like if we could slow down. Yeah. Somebody asks now, how quick can the occurrence cube be generated?
That's actually a very good question. And the answer is not so satisfactory. I think that depends. Because actually, by doing this, you are sending the SQL query to the GBIF cloud, to the GBIF compute nodes, and it needs time. So it depends on how many people in front of you are in the queue. And so that can range from a couple of minutes to a couple of hours. So that's really depending. In principle, most of your downloads that you will request from GBIF will be there within 15 minutes, I would say.
Okay. So there's a little bit of variance, but 15 minutes is just the time to go for coffee.
Yes.
Yeah.
And if that's too slow for you, then actually the GBIF dataset is also on Google Cloud, actually. It does require you to have some cloud credits to compute, but there is Google BigQuery, and this contains also the GBIF data. So you could also use BigQuery to generate your cubes.
Okay. Very interesting. Excellent. Thank you very much. Let me see. So there are two new messages. Okay, these are just thank yous and so on and so forth. Yeah. Excellent. Okay, if there are no, sorry, there are no raised hands. Yeah, no. Okay, then Maarten and Sandra, thank you very much. I'm sure that the participant will have the opportunity to check the slides, let's say by tomorrow, yeah? And if there are questions that come up during the week, they will find you next week in Zoom again. And we are using the same link next week as well. Yeah. So thank you very much. And yeah, I'm looking forward to the next edition. Yeah, thank you. Thank you very much,
Thanks,
everyone.
Francesco. Thanks, everyone.
Have a good afternoon. Nice photos behind you, Sandra.
Well, different parks.